Hello, everyone. Today, I want to talk a little bit about my research project, helping stress cells recover. But before I get into my work, let me just give you a little bit of background information. First things first, what even is cell stress? Well, generally, cells need to regulate their environment. Just like you can't get too cold or too hot without hurting yourself, cells also need to regulate not only temperature, but also nutrients, ions, and hormone levels. And cell stress is when something disturbs that balance. And when the cell becomes stressed, it often can't produce the parts that it needs to maintain itself, or it can't do its job. And if a cell can't do its job and it can't repair itself, then that cell's probably going to die, or at the very least become very damaged. And this, in turn, can make conditions like kidney failure or heart disease worse because you have fewer cells that it can actually function. So this is very obviously a bad thing that we want to figure out how to stop or reduce. So it can be caused by many different things from both inside and outside of the body. However, my research focuses more on the things that are outside the body or the environmental factors of cell stress. And these include starvation, which is the main focus of my work, but also lack of oxygen, calcium deficiency, which is a type of starvation, and then physical damage like cuts and temperature changes. So now that you have an idea of what cell starvation is, we can move on into my research project. So I am investigating the cell stress that is caused by starvation, and I'm doing this in order to try and find a way to minimize the damaging effects on the cells, because starvation is something that affects people all around the world, and this could help develop a treatment plan to help speed recovery from those who have suffered or are suffering from starvation. So the plant extract I'm using is called Angelica sinensis, and this plant has been shown to have many beneficial effects from anti-cancer properties to improving blood circulation. And because of its properties and how we think it interacts with the cells, it made a very good candidate to try and test to reduce the cell stress. And before we get into the actual experiment, I very quickly want to go over ICC. So ICC, or immunocytochemistry, is the method I use to take pictures of the different structures of my cells. It uses targeted antibodies that stick to very specific structures inside the cell, and then they'll glow a certain color under a microscope. So as you can see in the picture, the green is the skeleton of the cell, and the blue is the nucleus. So that's just a very basic rundown of what ICC is. So now we can go and jump into what I did for my project. So the very first thing I did was determine how the Angelica sinensis extract, so this is my plant extract, affected the structure of heart cells, because structure is very important to function. And if the structure isn't right, then those cells, they're not going to function properly. So if you look in the bottom of the slide, you can see that ICC image. So the green is the skeleton of the cell, and you can see how they're very long and skinny. And then if you look to the image to the right, you can see that they like to grow all in line with each other and very, very close to each other. And this is important because your heart needs to be able to contract in a very specific way. And so it needs to be tightly regulated all in the same direction in order to maintain its function. So as you can see to the left here in this ICC image, you have the starved cells. So these are the heart cells that have been denied nutrients, so they're starving. And you can see the structure has sort of degraded. It's gone from that long, skinny structure to sort of a blob. However, when you take your SARS cells and then add your Angelica sinensis extract, you see the restoration of that former structure. So they start to get skinny again, they start to realign, go all in the same direction. And if you compare to the heart cell from the previous slide, you can definitely see the difference between the ones on the left that are just starved with no treatment and the one on the right that are starved with treatment. And it's far more similar to the image on the right here. So that is very important and very good for my project because it shows that the extract can restore the structure of the starving cells to what it was like before they were starving. This is a good indicator that it can also restore function. So that was the first phase of my research, and this second phase is the stuff I'm currently working on. So I don't have a lot of images to show you guys, but we can talk a little bit about it. So I'm currently measuring how this plant extract affects cell death. So the available data so far indicates that the Angelica sinensis extract will actually decrease the amount of cell death that is present in these cells, and that's a good thing. We expect to see that. And I'm also looking at what genes are being turned on by the extract. And so far, it's looking like we have cell survival genes being turned on, which is, again, what we'd expect for something that increases cell survivability. And in the future, I want to identify the compounds and chemicals in the extract that determine what they are and then test them individually on the cells. And then based on the results from my current experiments, I would like to conduct, basically repeat them, but with different cell lines, because we also want to try lung cells, and then we have several different types of brain cells in the lab. So I want to investigate how the extract works in those cell lines, and then I also want to do it in live animals rather than in a dish to see how multiple different cell types interact with each other. So that was a very, very brief summary of my research project. And I hope you guys learned something today. And I'd like to thank you for listening.